Proper thawing is crucial for cell viability and the success of subsequent experiments. Careful attention to the thawing process ensures that cells resuscitate quickly and allows them to resume normal growth and function. Next, let's talk about how to thaw cells. Before you begin the thawing process, gather all the necessary materials. This will ensure a smooth and efficient workflow. When taking out the cryopreservation tube, take personal protection and wear gloves. Ensure that the operating table is clean and sterile, avoiding unnecessary pollution that endangers the health of cells. Prepare the experimental equipment to be used. Check the temperature of the water bath, the status of the centrifuge and centrifuge tubes, and sterile disposable gloves. Preheat the media to be used in advance. Having all these materials ready will minimize the time cells spend outside their optimal environment. Take the cryopreservation tube out of the frozen storage place and take it out quickly to avoid the influence of temperature difference on the cells. Immediately place the vial in a 37°C water bath to thaw. Cryovial can be thawed in sterile disposable gloves to avoid contamination. The step of thawing should be fast and should be completed within 1 minute, which is the key to recovering cells. Wipe the outside of the cryovial with 70% ethanol and transfer it into a sterile operating table. Slowly use a pipette gun to draw the liquid in the cryovial into a 15 milliliter centrifuge tube. Then add an appropriate amount of pre-warmed media and mix gently. Next is centrifugation. Centrifugation speed and duration vary by cell type. After centrifugation, discard the supernatant, add fresh media to resuspend the cells, and transfer them to a suitable culture container and culture environment. Be careful of the damage caused by low temperature when taking out the frozen cells, and wear anti-freeze gloves. It is very important to resuscitate the cells quickly. Try not to exceed 1 minute. Try to maintain high density after thawing to improve cell survival rate. Pay attention to aseptic operation and prevent cell contamination, which can help the cells to maintain a good state. The above is all about how to thaw cell. If you have any questions about this, please contact us.